When the average person thinks about a virus, it's usually because a member of his family is down with a disease such as measles, mumps, chickenpox, influenza, or poliomyelitis, or because the disease is unknown to the doctor and he is likely to attribute it to one of the still undiscovered viruses. Now, viruses were discovered just about 60 years ago, but most of the viruses that we know today have been discovered during the past 10 to 15 years. Now, although viruses are very important as disease-producing agents, and actually the viral infections of man represent the last great unconquered area of infectious disease, I believe they may be more important to scientists because of the approach which they may provide to a study of the evolutionary process by means of which all the myriads of kinds of life on this earth have been developed. I think they also may prove very useful to scientists as a means of studying about the nature of life itself. Now, viruses are too small to be seen by means of the ordinary microscope. But if you take a crystal of a virus, such as poliomyelitis virus, which we obtained in 1955, and slice it in two and look at it by means of the electron microscope, this is what you will see. Now, if you take one of those small round objects that you see there and enlarge it about 65 million times, it will appear such as the model that you see here. Now, if my head were enlarged to the same extent as this molecule of poliomyelitis virus, my head would be larger than the whole earth on which we live. Actually, you could get about 3 million of these little poliomyelitis virus molecules just on the pinpoint of a pin. Now, viruses come in different shapes and sizes. Here, for example, is a drawing of a bacterial virus, the same scale as this poliomyelitis model. It has a hexagonal head and long tail. And then over here, we have a model of a plant virus at the same scale. This model is of tobacco mosaic virus, and it shows about 1/10 of the entire molecule. If the molecule were all here, it would be about as tall as a 6-story building. Now, unlike bacteria, all of these viruses will grow only within living cells. And when they grow in these cells, they have the ability to cause disease. One unit of a virus, such as tobacco mosaic virus, in the proper place will cause disease. A unit of polio, for example, will cause disease and could cause death. Now, you may wonder, are there any good viruses, or are all viruses bad viruses? Well, actually, there are a few examples known where virus diseases can give pleasure to man. There are a few plants, such as tulips, for example, which, when infected with a virus or strains of viruses, will show beautiful colors and variegation. And because of this fact, such virus-infected tulips have been grown for many hundreds of years. If they had not had the beautiful colors due to a virus, they probably would not have been grown. So here, you can say that a virus might be regarded as being a good virus. Now, all of these viruses have another outstanding property, and that is that certain viruses will grow only in certain kinds of cells. Poliomyelitis virus, for example, will grow only within the cells of man and a few animals. It will not grow in plant cells. And a plant virus, like tobacco mosaic virus, will grow only in certain plant cells, and it will not grow in my cells or in the cells of animals. Well, now, to give you some idea of the place in which viruses fit into in the structure of all the kinds of structures that we have in the world, I'd like to show you a film which runs from very large structures, such as the whale, down to very small molecules of the chemist. We see the whale swimming in its natural habitat, water. These represent the extremes that we will now see in a series of structures each of which is about 10 times smaller than the other. Now, these living organisms represent the world of the biologist. And as you go down, you will eventually reach a very interesting area. Here you see a mouse, and as you go on down to a smaller structure, living structure, the mosquito, and then next a water flea, then still a smaller structure, the amoeba, and then still a smaller structure, the diatom, and then a structure still smaller by tenfold, a bacteria. And below that, 60 years ago, there was a blank. Biologists had smaller and smaller structures running down to the bacterium, below which they seemed to have a gap so far as structures were concerned. And then came the discovery of the viruses. And about all that was known of them was that they were still smaller than these accepted living organisms of the biologist. Now if we start at the other end of the scale, the chemist's world, and go from the water molecule to a larger molecule, the heptane molecule, all the way up to an amino acid molecule that you have in the proteins of your body, to a more complex structure, a B12, a vitamin, and next a protein known as myoglobin that we have in our bodies, and then finally a fibrinogen molecule that we have in our bloodstream. Now, the chemists then reached a same void or blank. This gap served quite well to separate the organisms of the biologist from the molecules of the chemist. It's obvious there's a tremendous difference between the smallest of the living organisms shown here in model form and the largest chemical molecule shown here. The gap between the two represents about 100-fold with respect to size. Now, just about the turn of the century, disease known as tobacco mosaic was found to be caused by an agent known as tobacco mosaic virus, which is shaped like a pencil. There followed the isolation of a whole series of structures shown here in model form. These represent the viruses. If we imagine a long line of ordinary inert chemical molecules arranged in order of size, the largest molecule proves to be a little bigger than the smallest of the viruses. The viruses range in size from this tiny, round tobacco necrosis virus up to the large smallpox virus. And this is just a little bigger than the smallest living organism, a model of which we have hung just above it. One can truly say that the twilight zone of life exists in this particular area, the overlapping with molecules on one end and with living organisms at the other end. Obviously, if you could find out the nature, the difference between a molecule which grows or replicates and a molecule which does not, you would find out quite a bit about the nature of life itself. Now, in order to give you a better idea of the tremendous difference between the molecules of the chemist and the living organisms of the biologist, I'd like to show you first a model of a chemical molecule. It is quite different from a living organism, which I'd like to show you in the form of the film. Here you see the organism moving about, the hairs or cilia making considerable amount of motion. You can discern an internal structure, and all in all, this living organism is tremendously different from the model of the molecule which you saw just a moment ago. Now let's see what happens when you take a solution of tobacco mosaic virus, shown here in model form, and apply it to a normal Turkish tobacco plant. This molecule springs into action and multiplies many, many millions of times over and causes disease. As you can see, by means of the film in which there has been a greatly speeded up action, you see first the clearing of the veins on the leaves. And then, as the disease develops, you see the beginning of spots, necrotic areas, dead areas. you begin to see some of the leaves are distorted a little bit. This is the tobacco mosaic disease resulting from the infection with a single unit of tobacco mosaic virus. Now, if you use still another type of host, you get a response in which the disease is localized and you get spots on the leaf. It's very important to recognize the fact that the number of these spots bears a relationship to the amount of virus that was used to rub over the surface of the leaf. It's important to be able to measure virus because if you want to know whether a virus is multiplying, you have to have some way of measuring it. Now, it is also possible to study other viruses, such as bacterial viruses, in a similar manner. If you have a layer of bacteria on a petri dish and pour over it some bacterial virus, it responds by having these little areas of dead bacterial cells known as spots or plaques. Now let's see how these develop by means of a time-lapse movie. You can see the layer of living bacterial cells over which the bacterial virus has been placed. You begin to see the spots forming. These spots are composed of dead bacterial cells. Now, in the same way, the number of spots that are produced there can be used to measure the amount of the bacterial virus. Now, I've indicated that viruses are very, very small. And in order to give you some idea of the relationship between the virus that you've seen here in model form and the disease and the virus within the cell, I'd like to have you look at a leaf coming closer and closer and closer. You see the leaf? And then as you come closer and closer to the leaf, you begin to see the cells on the surface of the leaf. These cells we call hair cells. Here you see a leaf on edge, and these hair cells stand out very beautifully. As you come closer to one of these hair cells in a diseased plant, you occasionally see a crystal. This crystal, shown in this upper cell, has been found to be composed of tobacco mosaic virus. If you dissolve up this crystal in a little water and look at this solution by means of the electron microscope, this is what you see. Now here you see the actual molecules of this virus. These are real life pictures. This picture was taken by means of the electron microscope. And Dr. Robley Williams has developed many of the techniques by means of which you can get such a beautiful picture of a single tobacco mosaic virus particle, such as shown here. We have relied heavily upon the electron microscope in virus research. Now I'd like to have Robley Williams tell you a little bit more about its use.
The electron microscope has proven particularly useful in discerning the shapes and sizes of virus particles. In fact, about all we know about their shapes and sizes has come from that instrument. Now, I'd like to take, however, another look at this tobacco mosaic virus particle to point out a couple of things. Actually, the electron microscope is rather powerful. For example, it's only a 1/2 millionth of an inch across this virus particle and about 100,000th of an inch along its length. But you'll notice that you do not see any particular detail of structure along its length. Now, in some cases, we can do a little bit better. For example, if we have a long thread or polymer, in this case of nucleic acid, we are able to discern it even though it's only 20 atomic diameters across. Now, the electron microscope does range from the large molecules of the chemists through the viruses and does particularly well at the range of size of the viruses. But it doesn't do well enough. What we really want to do is to see the small molecular dimensions of the virus particles in order to be able to understand better how they operate. Well, why not have the electron microscope magnified 10 million times? If we were to do that, we would find that we would destroy the specimen while we were photographing it, so we're stuck. So what do we do? What's done is to call in the aid of other people, and in this case, it's proven actually that the chemist and the X-ray crystallographer have been of as much use in discerning the detail shape, or at least guessing at it, of tobacco mosaic virus as the electron microscopist. And with the aid of all three of these people, it has been possible to make a sort of concept which one can make into a mechanical model of tobacco mosaic virus. This is magnified about 65 million times. One sees a spiral or helical array of protein subunits, and now I'm going to begin to take the virus apart by taking out one protein molecule at a time. These have molecular weights of about 17,000, and as I start removing them, we see that we can see something inside the virus. And what we see inside the virus is a rod. You will notice that the rod goes up the spiral following the pitch of the helix. It's only about 20 atomic diameters from here to here, but the rod is of nucleic acid, in this case ribonucleic acid, is able to follow the protein subunits all the way and essentially weld them together, leaving only a hole in the center of the virus. So our detailed concept, at any rate, of the structure of tobacco mosaic virus is that it's a great collection, some 3,000 protein subunits held together by the ribonucleic acid that spirals up and forms this beautiful structure which we see.
In contrast to the beautiful details of virus structure that you've heard from Robley Williams, it's difficult to realize that only 60 years ago, viruses were mysterious somethings, somewhat smaller than the smallest living organism, and yet with the ability to cause disease. The viruses were actually discovered first by a Russian botanist, Ivanatsky, but were first really identified by the Dutch botanist Beyering. Beyering took a mosaic disease plant, such as the one that you see here, and ground up the plant, pressed out the juice, and passed it through a filter which would hold back all living organisms. And he found that when this filtrate, the liquid that came through the filter, was applied to a plant, that it would cause the tobacco mosaic disease. He called this material a contagious living fluid. Certainly a contrast in terms, because how could a fluid, like water for example, be alive? Well, the true nature of viruses did not become known until about 1935, when after some years of considerable work, I had the good fortune to isolate this virus in the form of a crystallizable material. This turned out to be a compound composed of protein and nucleic acid. We call this a nucleoprotein. There were considerable doubts expressed in those days as to whether or not a crystalline material could actually be a virus, because in those days viruses were regarded as living agents. And how could a crystalline chemical actually be alive? Well, after a considerable period of controversy, many experiments were made which gradually built up the idea that these crystals really were tobacco mosaic virus. Now I'd like to create for you today that experiment that I made 25 years ago. If you take again a diseased plant such as this, only I used about a ton of these plants in the first experiment, and pass this juice first through a filter such as this, which will hold back large pigments and so forth, and then pass it through a so-called bacteria-proof filter such as this, which will hold back living organisms, and then you finally end up with a clear liquid, such as a liquid that you see here. I had a hunch that the material that remained might be protein in nature. And so I used a protein precipitating agent that the chemists use, an acid and some salt, and added it to this clear solution and watched to see what would happen. The clear liquid becomes turbid. Something has precipitated out. Well, I wondered about the nature of this, and so I decided to take a look under the ordinary microscope. I put a drop of this material on a slide, looked at it under the microscope, and here is what I saw. There are many, many long, needle-like crystals. Well, could this be tobacco mosaic virus? Well, I found by dissolving these crystals and diluting them out many, many thousands of times that the solution would cause the disease. And after much experimentation, I found that this crystalline material actually consisted of tobacco mosaic virus. Here is a little bottle containing some of the original crystals that I obtained in 1935. Each one of these needle-like crystals is composed of tens of thousands of the individual molecules that you saw just a moment ago. Now, it's possible to obtain viruses today in very large quantity. You see here a bottle containing a considerable amount of virus, the virus obtained from many hundreds of plants. Although a great many plant viruses have been obtained in crystalline form since 1935, no animal virus or a virus affecting human beings was obtained in crystalline form until 1955, when Doctors Schaefer and Schwert, working in the virus laboratory, were able to obtain poliomyelitis virus in crystalline form. Here is a little crystal of poliomyelitis virus. If you enlarge that crystal some 100 times, here is the way it will look. There is enough poliomyelitis virus in this crystal to cause poliomyelitis in every man, woman, and child in the United States if they were all susceptible and if the virus could be distributed in that form. Now, if you would slice through this crystal, such as you would slice through an apple or an orange, and look at the slice of the crystal, here is what you would see. Each one of these particles of poliomyelitis virus has the ability, if it gets into a susceptible cell, of causing disease. Now, if you enlarge one of those particles of poliomyelitis virus some 70 times, you obtain something similar to this. This model of a single molecule of poliomyelitis virus is actually enlarged about 65 million times. This model represents just about all we know of this molecule from information obtained by means of the electron microscope and by means of X-rays. Now, these knobs that you see here are protein subunits. We believe there are about 60 of these around the outside of this particle, and in some cases, these protein subunits are arranged in the form of a pentagon. Other viruses have still other shapes. For example, here is a large-scale drawing at the same scale of a bacterial virus. It has this large hexagonal head. It has the nucleic acid inside, and this runs down through a long tail, extending all the way down to the far end in some tail fibers, by means of which this virus actually attaches itself to the host. And still another virus, of course, is our old friend tobacco mosaic virus, the model that you saw a few moments ago, with the protein subunits on the periphery and the nucleic acid running round and round up through the center of this. Now, this model, as I have indicated, is only about 1/10 the true size of this molecule, and if it were extended to full size, it would be about as tall as a 6-story building. Now, all of these diverse structures, poliomyelitis, tobacco mosaic, the bacterial viruses, all have one thing in common. They all possess the ability to cause disease when they get into the cells in which they can multiply. When they do this, they seem to have the ability to take over the metabolic machinery of these cells. And the cells, instead of making the normal proteins and the normal things that we need for a good life and a healthy life, they cause the cells to make more of these big structures that you see here, of these virus particles. And as the cell makes hundreds of thousands of these viruses, the cell becomes diseased, and we then become sick. Now, when a virus, one of those virus particles, gets into a cell, we really don't know how it takes over this metabolic machinery. This represents one of the very great problems that's available for study at the present time. Now, during future programs, we hope to tell you much more about these viruses. Perhaps a little bit more about the metabolic machinery inside of virus-infected cells. We hope to tell you a little bit about these viruses as giant molecules. And then we will tell you more about some of the diseases that these viruses cause. And then there's a very important area because, as I've indicated, these viruses are very similar to genes or chromosomes. They are made up in part of nucleic acid. Now, nucleic acid was discovered a long time ago because it occurs in the nuclei of all of our cells. Well, as you now know, it also occurs in the viruses. And Robley Williams showed you an electron micrograph of one of these nucleic acids, and you remember it was a very long strand. Now, it's actually composed of several building blocks, and the actual nature of the message that is used to transfer information from parent to child, and which results in eye color and in the color of hair and so forth, is written in this nucleic acid. Now, if we could learn more about the nature of the nucleic acid, we'd have a much better idea concerning the way that all of life has evolved on Earth. Another problem, of course, is the problem of whether or not we at some time will be able to synthesize one of these viruses in the laboratory. You remember that a virus, in some respects, is like a living thing. And obviously, if you could synthesize some possessing the properties of life, it would represent a tremendous achievement and at the same time, a tremendous problem for society.
Virus was produced for the National Educational Television and Radio Center by KQED San Francisco, in cooperation with the Virus Laboratory of the University of California at Berkeley. Featured in this program were Dr. Wendell M. Stanley, Professor of Virology and Director of the Virus Laboratory, and Dr. Robley C. Williams, Professor of Virology and Associate Director of the Laboratory. Next week, Dr. Williams will discuss viruses as giant molecules and describe how we've been able to measure, weigh, and even dissect them. This series was made possible by a grant from the National Science Foundation. This is National Educational Television.